Now time to look at cellular reproduction and protein synthesis, both major functions that occur inside our cells. The whole purpose of cellular reproduction is to reproduce more cells. So one of the functions of a living thing is to be able to reproduce, and so this is what allows it to happen. Inside our body, at any one time we have trillions of cells, but those cells are there are cells that are constantly dying, and then there are new cells being created, and this is how it happens in what's called the cell life cycle. So if you take a cell's life, you can split it into two major parts. And the first one we call interphase. This is the majority, the longest part of the cell cycle, up to around 90%. And during interphase, this is when the cell is doing its normal everyday life things. So during interphase, the cell is going to grow. It's going to carry on any metabolic processes. If it's a skin cell, it's going to do skin cell things. If it's a heart cell, it's going to be pumping blood and doing heart cell things. That's all happening during interphase. And then we have the cell division. This is where the cell is going to replicate itself. It's the rest of that 10% of the cell life cycle. And its job is to, in us, the reason we have cellular division is for growth and also for repair. So as we have gotten larger over our life, started as a very small baby, now as an adult, the reason we're able to grow is because we've been producing more cells. And then if you were to cut your cell, that wound is going to heal over, and that's also brought about by this cell division. So the parts of the cell division are as follows. All of these parts, we have a name, we call this mitosis. Mitosis is another name for cell division. And it's been split into four major stages: prophase, metaphase, anaphase, and telophase. And notice here that in mitosis, we're starting with one cell, and we're ending up with two cells that are identical to that mother cell. They're identical in their DNA, they have the same DNA, etc., etc. So if you think about it, we all started as one single cell. After fertilization, as the egg and the sperm came together, we were a single cell. And so this cell division process has happened millions of times inside our body, and it's continually happening every day as we've gone from this single cell up to this multi-celled organism that we are now. Then there's also something called cytokinesis, and this is the actual breaking apart or division of the two cells into one. So during these four stages of mitosis, prophase, metaphase, anaphase, telophase, we're mainly going to focus on what's happening with the DNA. So remember, the DNA is the information in the cell, and so it makes sense that in cell division we would focus on what's happening with the DNA. Cytokinesis, this is where we're splitting apart the cell. So let's look at each of the phases here. Here's a cell in interphase. So notice it has a single nucleus, we've seen before, plasma membrane. Here are the centrioles. I said before that the centrioles would become important when we talked about cell division. The centrioles, as we'll see, are important in creating what are called spindle fibers, which move the DNA from one cell to the two new cells. At this point, the DNA is still what we call chromatin. Chromatin is DNA that it's not condensed. It's kind of strung out into this thread-like structure, and it's found spread out throughout the nucleus. After interphase, we're going to now enter into mitosis. So now we're into mitosis. Once we pass this spot right here, we've now left interphase. Now we're entering cell division. Notice here that we now have what are called chromosomes. Chromosomes are also DNA, but they are a condensed version of DNA. So notice here in this nucleus here, the DNA is there, but it's in the form of chromatin. We can't really see it. When that DNA condenses down into chromosomes, we can see that under the microscope. And this, when we think about chromosomes, we think of this little X-shaped figure, something like this. And that's what a chromosome looks like, and it's made up of what are called two sister chromatids. So this side of the DNA over here is one sister chromatid. Here's the other sister chromatid, and these sister chromatids are identical to each other. So at this point, the DNA has already duplicated. So one of the major things that has to happen in mitosis, if we're going to go from one cell to two cells with the exact same DNA, that DNA has to duplicate. That's already happened when we can see the chromosomes. Notice up here we have what are called spindle fibers starting to form. So here's our two centrioles. And from these centrioles are coming these spindle fibers. And they'll become important here in just a minute. But you can see they're kind of like long strings. They look almost like a spider's web or something like that. Here's also the centromere. There's lots of C words in this part of the lecture, but a centromere is the center part of a chromosome where the two sister chromatids are attached together. So in prophase, we have the chromosomes condensing, we have the spindle fibers starting to form, and then the other thing that's going to happen in prophase is our nucleus is going to disappear. So we have fragments of the nuclear envelope. So the DNA is usually found in the nucleus. The only time the DNA ever leaves the nucleus is during cellular division. Other than that, if you want to find the DNA, it's always going to be in the nucleus. But in order to move that DNA to two separate cells, we have to get rid of that nucleus just for a short period of time here. Notice these spindle fibers are now completing their job. And what they're doing is they are attaching to the chromosomes at the centromeres. And at this point, the spindle fibers are just attaching on one side. But here in a minute, we'll see that the spindle fiber is going to attach the chromosome from each side of the cell. Those spindle fibers are what are going to move those chromosomes around. So now we have finished prophase. We're now going to move into metaphase. This is the second step. During metaphase, notice the chromosomes have all lined up in the middle of the cells. There's a spindle fiber from this side attached here, spindle fiber from this side, and between prophase and metaphase, there's this little tug-of-war going on, and the spindle fibers are pulling one way and then the other way until finally all the chromosomes are lined up in the center of the cell. And that's when we know when we've reached metaphase. And that's the main part that happens in metaphase. Then we move into anaphase. Now in anaphase, the spindle fibers are going to pull the chromosomes apart. Remember, the chromosomes are made up of two sister chromatids, identical halves, two identical halves. So in anaphase, the spindle fibers are going to start shortening, and as they shorten, they're going to pull those two sister chromatids apart, so that going this way, we have half of the DNA, and this way, half the DNA, but this sister chromatid here and this sister chromatid here are identical, so this side of the cell is going to get one copy of the DNA, and this cell is going to get the other copy of the DNA. And then we reach telophase. In telophase, basically everything that happened in prophase is going to unhappen. So remember, in prophase, first of all, the chromosomes condense. So between anaphase and telophase, all of the chromosomes have moved to one side or the other. So we have one copy of the DNA here and one copy here, exact copies of the DNA on each side. And so in telophase, these chromosomes are going to decondense back to chromatin. Also, the spindle fibers formed in prophase. So in telophase, the spindle fibers are going to disappear. And then also in prophase, the nucleus disintegrated, fragmented. So in telophase, the nucleus is going to reform. And then we have what's called cytokinesis again. And cytokinesis is right here, where these two cells are going to pinch in half. So the way our cells split, it's almost like taking a balloon, and if you could pinch that balloon in the middle, and pinch it so tightly that eventually it popped into two balloons, and they'd still be whole balloons. Or maybe like taking a piece of gum. If you take that piece of gum and stretch it, so that middle gets thinner and thinner and thinner until finally it breaks apart. That's what happens in our cells. That's cytokinesis. So notice that these two cells, you get what's called a cleavage furrow. And then this is going to shrink and it's going to come together here and here until eventually we're going to end up with one cell over here with its copy of the DNA, and the other cell over here, they'll no longer be attached with its copy of DNA. And there we go. Then we have two cells where we started with one. That is cellular reproduction. I'd like you to know the major stages here and what's happening in each. So just to recap, prophase, the chromosomes condense, the spindle fibers form, the nucleus disappears, and those spindle fibers attach to the chromosomes. Metaphase, the chromosomes line up in the middle. In anaphase, the chromosomes are pulled apart so that half the chromosome goes, one sister chromatid goes one side, the other goes the other. Then in telophase, the nucleus reappears, spindle fibers disappear, the chromosomes decondense into chromatin, and then we have cytokinesis occurring. Again, this is going on constantly inside our body. The process we want to talk about here is something called protein synthesis. This is a pretty complicated process. I don't want to spend a lot of time on this, but I do want to point out to you how proteins are built, because proteins, again, they're the workhorse of the cell. They do a lot of things, and so protein synthesis is a very important part of cellular life. So, a few things to point out here. We've already talked about cells. And inside the cell, we've talked about the DNA, and the DNA is housed inside the nucleus. We've also talked about ribosomes, which are these little organelles, these little structures, maybe not necessarily organelles, but they're these little structures outside of the nucleus that synthesize proteins. Now the DNA, interesting thing about DNA, we said that it holds the information. The information that it holds is the information to build proteins. That's what the DNA houses. That's the information in the DNA. So the information in the DNA is used to make these proteins at the ribosomes. Problem, though, is that DNA never leaves the nucleus. So the DNA is always found inside the nucleus. Protein synthesis, however, happens inside the cytoplasm at the ribosomes. So how does this information get from the DNA to the ribosomes? This is where RNA comes in. RNA is the messenger between the ribosomes and the DNA, because RNA can be in the nucleus, but it can also leave the nucleus and be found in the cytoplasm. So the way this works is we go from DNA, and a copy of the DNA is made in the form of RNA, and then that RNA is read, and that information is used to make our proteins. And this is happening in every cell of our body all the time to produce proteins. Proteins can be enzymes, they can be cytoskeleton, we mentioned before. Skeleton is all made up of proteins. Antibodies are proteins. Carriers or channels in the plasma membrane, all of these things are proteins. Very important type of organic molecule. So let's look briefly at protein synthesis. There's a few different types of RNA that are all used in protein synthesis. One of them is called messenger RNA, and this is the one that is a copy of the DNA that goes between the nucleus to the cytoplasm. There's also something called rRNA or ribosomal RNA, and it is used to form ribosomes. So ribosomes are made of RNA and also some protein. Remember, the ribosomes are where proteins are built. Then there's also something called transfer RNA, and transfer RNA's job is to bring the amino acids which build proteins. Amino acids are the building blocks of proteins. So transfer RNA reads the information on the messenger RNA and then goes and gets the appropriate amino acid and brings it back to the ribosome in order to build that protein. This whole process happens in two steps. The first is called transcription, and in transcription we are simply going from DNA to RNA. Transcription happens inside the nucleus. Then there's the second process, which is called translation. Translation is the process of going from RNA to proteins. And this happens in the cytoplasm at the ribosomes. So let's look briefly at a picture here at how this happens. So what we have in this picture, the purple is the nucleus. Inside the nucleus we have the DNA. The DNA is this purple double-stranded structure here. And then we have this blue stripe here, this ribbon-like structure here. That is mRNA. So notice that the mRNA here is attaching to the DNA. The mRNA is actually being built at this point. So the mRNA is making a copy of the DNA. So the DNA is not going to leave the nucleus, but the RNA is, and it's going to go out into the cytoplasm here. So notice now we have this blue ribbon. It has now left the nucleus and it's come out into the cytoplasm, and it's found a ribosome. So this red guy here is a ribosome. So the mRNA is going to attach to the ribosome, and then the sequence of nucleotides. So remember, nucleotides build nucleic acids. So both DNA and RNA are built of nucleotides. These nucleotide sequences are going to help determine what protein is going to be built. So we have our copy of the DNA comes out into the cytoplasm, attaches to a ribosome, and then we have our tRNA here. So this little guy here is a tRNA, and these are our amino acids. So the tRNA is going to pick up an amino acid depending on the sequence of nucleotides here in the ribosome. It picks up the appropriate amino acid and brings it back to the mRNA. So notice here the tRNA is bringing this amino acid to start building the protein. And then what's going to happen is, as the mRNA is read by the ribosome, the tRNA is going to bring these amino acids and form this long chain of amino acids, which is our protein. So this section here is called transcription. In transcription, we're going from DNA to RNA. In the cytoplasm here, we have translation. So in translation, we have the mRNA, the rRNA, and the tRNA coming together to form our end product, which is this, our protein, our amino acid sequence. And that's the whole purpose of this whole process is going on here. Once that protein is formed, it can then be modified and it'll go on to do whatever function it is, whether it's a hormone, whether it's a part of the cytoskeleton, it'll be now ready to go and do its job. So that is now cellular division and protein synthesis.